Today I'd like to talk to you about automated reanalysis in the field of genomic medicine.
But first, let's talk about what DNA is. DNA contains the instructions that tell our bodies how to grow and develop. Each person has a unique DNA sequence that affects many things about them, such as their eye and hair color, as well as their chance of developing some diseases. DNA is made up of a sequence of letters, A's, T's, C's, and G's. So what is genomic sequencing? Genomic sequencing is a technique that is used to read DNA. It finds the order of the letters of a person's DNA one by one. This DNA sequence can be referred to as genomic data. There are a lot of letters to be read, which means that genomic sequencing can produce a lot of data. Once the DNA sequence is obtained, the genomic data can be used by doctors and scientists to help identify the causes of genetic disease in patients. Doctors and scientists look for changes in the DNA, called variants, that are different between people with diseases and people who are healthy. These changes might be very small, like spelling mistakes, or bigger, where large runs of letters are missing.
But what happens if genomic sequencing doesn't find a diagnosis? Well, in Australia, from 2018 to 2021, around 25,000 people had a genomic test. Of these, 17,000 were unsolved, meaning the patient didn't get a diagnosis. We know that 10 to 15% of unsolved cases will be diagnosed every couple of years if their data was reanalyzed in light of new knowledge and analysis methods. 900 of these have been reanalyzed to date, but that's only 5% of the total that need reanalyzing. This means that at the current rate, using human power and manual analysis alone, it will take 85 years to reanalyze these tests. That's 85 years before some of these patients get a diagnosis. So why aren't samples being reanalyzed? One of the main things preventing reanalysis from taking place is the fact that reanalysis of genomic sequencing data is currently performed manually by laboratories on a case-by-case basis, with reports issued back to requesting clinicians. Unfortunately, laboratories don't have the person power to increase the amount of reanalysis they are doing. There is also very limited funding. Reanalysis is currently funded in an ad hoc manner through clinical services from existing budgets. Recently, Medicare funding became available, for example, for those children who have had Medicare-funded testing for syndromic and non-syndromic intellectual disability. Medicare will fund 2 cycles of reanalysis, at least 18 months apart, up to the age of 15 years. So what can we do to try to solve this massive problem? We are proposing to automate the process of reanalysis. This is expected to benefit thousands of patients that do not have a diagnosis. This automated reanalysis process will be designed so that undiagnosed cases are automatically reanalyzed continuously without requiring clinician-initiated requests. Highly suspicious variants flagged by the automated reanalysis program will be formally assessed by diagnostic laboratories, and disease-causing or likely disease-causing variants will be reported to the original requesting clinicians.
Thanks for listening, and we look forward to seeing you in the focus group.